Okay, so welcome back to this next video in which we are discussing cancer development. Okay, right, so we're currently in the process of discussing a simplified picture of cancer development. Okay, so we've discussed that what's going to start the whole process off is a single cell is going to get some change, which is either going to be a genetic change, a mutation, or an epigenetic change, okay, which might be caused by, for instance, carcinogenic chemicals, or potentially ionizing radiation or ultraviolet radiation, but which also could just occur because the cell makes a mistake. Particularly in dividing cells, you are going to get a huge number of mutations occurring in dividing cells every time a cell divides, okay, because DNA replication is not flawless, and some of these mistakes are not picked up by DNA repair mechanisms, okay, and we know that cells are not completely not dividing in the adult human, okay. We have these epidermal stem cells and their later cells, for instance in the skin and the intestine, which are dividing. So we saw the transit amplifying cells in the epidermis, those were capable of dividing, okay, so you do have division in the adult human. In addition, that second class of terminally differentiated cells, okay, I think I've still got it on this piece of paper, yes. These cells, so for instance fibroblasts, smooth muscle cells, and the epithelial cells of the liver, the pancreas, the lung, the prostate, and the breast, although those don't generally divide, they can divide, okay, if necessary. So you might be getting division, okay, under certain circumstances, okay. So you do have cell division in the adult human, and this can alone lead to mutations. Okay, right. So some sort of change has occurred in this cell up here, and what this change is now going to lead to is this cell completely overdividing. Okay, now we're not going to go into the specific signaling pathways within the cell, okay, which can actually go wrong, okay. We're just going to do this broad overview of this topic, okay. So broadly, what can inside cells you have two different types of signaling pathway, okay. You have signaling pathways which are pro-growth, okay, which are pro-growth and division, okay, so let's put this in here. So certain signaling pathways are going to be pro-growth and pro-division, okay, whereas there are other signaling pathways which are anti-growth and anti-division, okay, and cells are all the time getting signals that are either telling them to divide or telling them not to divide, basically, and the cell has to weigh up which of these signals is stronger and decide whether it should do anything or not, okay. So if you want to get a cell to overdivide, either you can have a gain of function in the pro-growth and pro-division signaling pathways, which lead to this signaling pathway being overactive in this cell unnecessarily, okay. So some sort of mutation that leads to these pro-growth and division signaling pathways being overrepresented in this cell, badly overrepresented, okay. So they shouldn't be being activated, but they are being activated to a far greater extent than they should be, okay. And there are many different ways that you can do that, either by actually having mutations in the proteins in these signaling pathways, or potentially by actually overproducing the signaling molecule which activates these pathways in the first place, okay. Or you can also overdivide by losing function, so loss of function, and there are nice abbreviations for this. Gain of function is GOF, loss of function is LOF. Okay, so you can either gain function in the pro-growth and division signaling pathways, okay, or you can lose function in the anti-growth and division pathways. Okay, and if you lose function in the anti-growth and division pathways, then once these are underrepresented, the cell will only have the small amount of stimulation of the pro-growth and division pathways, and therefore the balance will be tipped anyway, okay, so the cell will then overdivide. So both of these can lead to problems, basically mutations in both of them. Okay, so you can mess around with this in two ways, basically. Okay, so the cell is going to get some sort of problem that then leads to it over-dividing. So now what's going to happen is this first initial cell is now going to over-divide. So let's put this here. So here is our duct once again. Okay, and now what you're going to start producing is a new population of these cells. Okay, so now the duct is being obscured slightly. Okay, so here are the normal cells. So these are the cells that weren't affected. Okay, so these are the nice duct cells. And now, let's say this was the orange, this was where the original orange cell was, and now it's divided and divided and divided and produced a whole new population of cells. Okay, so I'll put these here. Okay, and these cells are no longer all attached to the basal lamina of the duct anymore. Okay, so here is the basement membrane or the basal lamina of our duct here. Okay, and here then in orange are all the cells which have come from this one aberrant cell here. So let's say that these two surrounding here are also aberrant cells, and then all of these ones that are stacked on top here, these are all progeny of these aberrant cells. Okay, so why has this happened? Well, basically what's happened is when the cell got this heritable change that meant that it over-divided, okay, whatever that change is, and we're not going to go into the molecular biology of what actually that change could be, okay, a lot is known about what that change could be, but we're not going to go there, not in this video, okay. What's going to happen is the cell will divide and produce two new cells, okay, and the key thing is that now both of these progeny cells, both of these daughter cells, have also got that change that leads to them over-dividing, because I said that this change had to be heritable. So now both of these daughter cells will over-divide, okay, and you'll go from 2 cells to then 4 cells, okay, and then from 4 cells to 8 cells. So before long you will have a huge great population of these cells which are all identical and which have all got this same problem that means that they are now over-dividing. Okay, and this is, by the way, why this is often called a microevolutionary process. So people often refer to cancer development as a microevolutionary process, or if they're being even more fancy, they might call it a Darwinian-like microevolutionary process. I'll put this in here. So a Darwinian-like, okay. I don't know why it's quite Darwinian-like, because it is Darwinian evolution, really. Okay, so what does this mean then? Why is it called this? Well, firstly, let me start off with what microevolutionary means. So basically, hopefully you know what evolution is. It's the gradual change in the characteristics displayed by a population over time to favour characteristics that better enable that organism to survive in the environment. Okay, now in the case of human beings, evolution takes years and years and years. In the case of multicellular organisms, it takes years and years and years and years, basically, because humans reproduce on such a slow timescale. Microevolution refers to much quicker evolution, okay? Evolution on really short timescales. Now, when can microevolution occur? Well, when reproduction occurs on a really short timescale. So basically, when you see evolution in bacterial populations, that will be referred to as microevolution. Now, often people would just call it evolution, but you can call it microevolution if you want. And often cancer, for some reason, is called microevolution because it occurs on the timescale of a human lifetime. So basically, when you see bacterial resistance arising in a bacterial population to an antibiotic, that's a microevolutionary process. Okay, cancer, why is it called that? Well, basically it's because these cells are now better able to survive in the environment. These cells are doing better than these cells. Okay, then these cells are awful. Okay, they're awful for the multicellular organism, but actually when you view these individual cells as little individual lives in their own right, as organisms in their own right, these are actually doing fantastically well. Okay, it's produced a whole population of cells that are genetically identical to it. And note, these cells are now not the same as these cells. They have got this potentially a mutation, or maybe an epigenetic change. To keep it simple, keep in mind mutations. It usually is mutations, but these epigenetic changes in chromatin structure, are important too. Okay, so basically these cells are now different from these ones, and they've made a population, okay, and they're basically out-competing these other ones. They are doing better, okay, and this is why cancer is often called a microevolutionary process. It's a process that is occurring within the cells, okay, and it's a gradual change in the phenotype of these cells over time that better enables them to survive. Okay, you have to understand that the cells are not foreseeing what's going to happen to the human. Okay, yes, they're going to kill the multicellular organism in time, and over time, therefore, they're going to destroy themselves. But in the short term, these cells are doing fantastically for themselves because they're creating a bigger population, which is why it's often compared to a Darwinian process of evolution. Okay, basically, these cells are better at surviving than the other cells. Okay, the other cells are better for the survival of the multicellular organism, but these cells are better at surviving as individual cells. And that's why at the start I said that really cancer is the return from cells which are behaving in this very socially responsible way and sacrifice themselves for the multicellular organism. And it's the return of these sort of primitive bacterial cell-like behavior in these human cells. Okay. And really, that's what we're returning to. The cells are no longer sort of behaving in a way that is good for the survival of the multicellular organism. Now they're behaving in a way that is better for their own survival. Okay, right. So that's the first step. Okay, now in reality, we're going to make this picture more complicated in a moment, but in my first basic picture, I am now going to call this structure that we have here a tumor. Okay, so this is now a tumor. Okay, also known as a neoplasm. So the other name for tumor is neoplasm. Neoplasm is a slightly more fancy name. Okay, now neoplasm literally means new growth. So neo means new, and then plasm means growth. Okay, so this is a new growth. Right, now at the moment this is not cancer, and that's important to understand. Okay, this is a benign tumour. Okay, in order to be cancer, it has to gain another very important property, and it has to become what's known as a malignant tumour. So at the moment, this structure that we have here is known as a benign tumour. Now, benign tumours, I'm afraid, often go on to become malignant tumours, so they are not so innate, they're not so inert. Benign means harmless, basically. In reality, benign tumours are often not harmless, because they can often go on to become cancer. And also, benign tumours in themselves can be disease-causing, okay, because, these things can obstruct passageways. This could obstruct this duct, and therefore it could cause pathology through its mere presence, basically, even though it's not yet cancerous. Okay, right, so let's now talk about how a benign tumour is going to become a malignant tumour. So what is the difference between a benign tumour and a malignant tumour? Okay, so a malignant tumour has to contain cells which are invasive. Okay, now what does invasive mean? It means that these cells are not static, basically. They move around, okay? So the cells of this tumour that I've shown you previously, so I'll draw the picture out again on this page. So here is our duct, here are our duct cells, and here then is this mass of cells that we have now grown from that first aberrant cell here. So here are our normal, healthy duct cells, and then we've got our benign tumour here produced from this one original aberrant cell here. Okay, so I'll divide this up here. Right, so I'll colour all of these in orange here. So basically, at the moment, all of these cells in orange here, they are static. They're not moving around. They can't move around yet. Okay? So you have produced a tumour because all of these cells are over-dividing, but they are at least not moving. They're not going anywhere. Okay? The key property of malignant tumour cells is that not only do they divide ridiculously quickly, but they also move around. Okay, so that's what invasive really means. It means that they are capable of moving around too much. Okay, right. So, why do benign tumours lead to malignant tumours? Well, basically, in order for you to become a malignant tumour, you now need to get another mutation. Okay, and the reason that benign tumours tend to lead to malignant tumours is that this is now an utterly ideal breeding ground for malignant cancer cells. Okay, you have a huge number of cells here, all of which are capable of dividing very, very fast. Okay, if you were to if one of these cells gets a mutation that means that it can now move around, what's going to happen? Okay, let's imagine this. Let's say that this cell that I'm now going to overcolour in purple here, I don't know if that's shown up at all, okay, that cell there in purple, that cell is now going to get another mutation, okay, so it's going to undergo a further mutation or an epigenetic change. It's going to undergo another heritable change that is going to allow it to move around. Now, what sort of changes would lead you to move around more? Well, to understand that, you need to understand what usually stops cells from moving around. Well, the reason that cells are not capable of moving around normally is that cells have connections between themselves and other cells, and also between themselves and the extracellular matrix. So let's just quickly discuss this. So, basically, cells have molecules on their surface, okay, known as cell adhesion molecules, which can interact with cell adhesion molecules on the surface of other cells. Okay, so this is what's known as a cell adhesion molecule. Okay, and for short, cell adhesion molecules are often abbreviated as CAMs, for short CAMs. Okay, so cell adhesion molecules are involved in attaching cells where they are and stopping them from moving around, and they do this in two ways. The first, is that you can have these direct cell-cell interactions, as they are known. Okay, so you have a cell adhesion molecule on one cell, a cell adhesion molecule on cell 2, okay, and these two cell adhesion molecules then bind together to create what's known as a cell-cell interaction. Okay, and I want to stress there are a huge number of different cell adhesion molecules. Okay, there is not just one cell adhesion molecule. There are a huge number of different cell adhesion molecules. Okay, so there are a huge number of different molecules which are capable of binding two cells together in this way. Okay, now cell adhesion molecules also do other things. Okay, the other way that they can adhere two cells together is a more indirect way. Okay, so this is what's known as a direct interaction between two cells. Okay, you have two cell adhesion molecules on the surface of the two cells which bind together. Okay, then the more indirect way is that you have your two cells here. Okay, both of them have cell adhesion molecules on their surface here, but instead what's going to happen is the cell adhesion molecules are going to attach to components of the extracellular matrix. Okay, so remember, the gaps in between cells, the extracellular fluid is not just a gloop, okay? Instead, the extracellular fluid contains a meshwork of protein filaments and huge carbohydrate fibers, okay? And this is a very rigid structure that spans the gaps in between cells. Now, cell adhesion molecules on the surface of cells can attach to these components of the extracellular matrix. So this is some component of the extracellular matrix, and for short, the extracellular matrix is often abbreviated as ECM for extracellular and then matrix. Okay, and this is what's known as a cell matrix cell interaction. Okay, so these sort of interactions using cell adhesion molecules, cell-cell interactions and cell matrix interactions, these both hold cells where they are, basically. Okay, so if you want to no longer stay put, basically, you need some sort of mutation that is going to stop these sort of interactions. Now, it's not very well understood how this occurs, okay, because there are an absolutely huge number of cell adhesion molecules, so you would have thought you would need a lot of mutations to get complete and utter remission from cell-cell interactions and cell matrix cell interactions, but somehow cells do manage it. And as I say, it's not well understood at all how this happens, but some sort of change happens that means that these interactions are lost, okay, and these cells now are free to move around as they like, basically. Okay, and now what happens, okay, is, so we've started off with this single cell in this orange population that somehow got this change, and now what happens? Well, it's also got the mutation that it got from the original orange cell, which was that it divided too fast. So now this cell is going to divide really, really rapidly, okay, and it's going to produce a bunch of cells that all divide very, very rapidly, and also that all have this problem of not staying put, basically, of being motile. So what you're now going to get, okay, is if I show this, here is our duct, and now you're going to have these cells going all over the place, basically. So let me show this. So here are our normal duct cells still. Now we've still got our tumour here, okay, but now it's breaking out, basically. Okay, and it's these pink cells that are breaking out. Okay, so let's firstly add on the basal lamina here in turquoise. Okay, and then we'll put on the normal cells of the duct, which are sitting on the basal lamina here. Okay, and then we've got our tumour here, which consisted of now both orange cells, okay, which are the cells which just have the mutation that mean that they overdivide, and now we've also got in amongst those orange cells a mixture of these purple cells, and these purple cells, not only do they overdivide, but they also are incredibly motile. Okay, so let's colour these in. So all of these ones that are extending out of the duct now, they've broken through the basal lamina, and I'm sorry, it's smudged a little bit there, and they're breaking out into the surrounding tissue. They're definitely purple cells. Okay, but you're also going to have some purple cells in the original tumour. And then we'll also have some orange cells in our original tumour here. Okay, so here are our orange cells here. Okay, and I really should have waited more time before adding on the colours. Okay, there we go. It's smudged horribly, but never mind. Right, so this is an important concept, that in cancer cells you have cells which are different, in tumours you have cells which are different at different stages along the progression towards cancer. These orange cells were further back in the progression towards cancer, and now these purple cells are further along, basically. Okay, and this is the concept of intra-tumour heterogeneity. So intra means within, so within tumour, heterogeneity, hetero means different, okay, so these are cells which have got different genetic phenotypes, basically, okay, and these cells here, these purple ones, are different from the orange ones. So that's the concept of intra-tumour heterogeneity, that within a tumour you can have these genetically different cells, okay, which are different ways along the progression towards cancer. Okay, right, now these purple cells, these are the true cancer cells. Okay, this tumour is now called a malignant tumour. Once you have got cells that are invasive in the way that these purple cells are, that then is a malignant tumour. Okay, and it's these invasive cells which are overdividing, which are cancer cells. Okay, right. Now, what's going to happen now is that these cells are going to invade blood vessels. Okay, they're not just going to invade the surrounding tissue, they're going to get into blood vessels. Okay, well actually first I should probably say that they're going to invade the surrounding tissue. So they're going to invade the surrounding tissue, they're often going to end up destroying the surrounding tissue, and then they're going to find their way into two types of vessel. Okay, the first is they're going to find their way into blood vessels, but also they're going to find their way into lymphatic vessels as well. Okay, so we'll start with blood vessels. So let's say this is some capillary here. Okay, as this tumour grows, what's going to happen is that it's just going to reach the capillary and it's going to continue on and it will break into the capillary basically. So let's sort of put this here. So here it is, breaking into the capillary there. Okay, and now these cells, remember, they're incredibly invasive. They're not attached. They're very mobile. Okay, so they're just going to end up getting brushed into the blood basically and carried around in the blood. Okay, so what you're now going to have is some of these tumour cells are just going to end up going into the blood. Okay, like so. And they generally go in as single cells. Okay, so you don't get clumps of cells moving around in the blood. You get single cells being given off into the blood. Okay, so these cancer cells that are now in the blood are known as circulating tumour cells. Okay, and for short, circulating tumour cells are often abbreviated to CTCs. So C for circulating, T for tumour, and C for cells. So circulating tumour cells. Okay, right. Now, what can then happen is they can end up getting stuck to the blood vessels at other sites. So these tumour cells can go far and wide, they can go round the circulation, and then what can happen is that they can go to some totally different site, far away from where the initial tumour was. Okay, and here's this blood vessel at this far distant site here, and what can happen is the tumour cell can get stuck to the epithelium or at this far away distant tissue. So here is our tumour cell now stuck here. Okay, and then what will happen is it will start setting up another tumour here. It will divide out of control, like so. It starts invading into this new site here, and then you are getting the formation of a new tumour far away from where the initial tumour was. Okay, now there's some nomenclature that's used here. The initial tumour is known as the primary tumour. Okay, so this one here, where the cancer actually started, this is known as the primary tumour. Okay, and here, where this new tumour is being formed, this is known as the secondary tumour. Okay, now secondary tumours are also known as metastases. Okay, so a secondary tumour is called a metastasis. So metastases is the plural, metastasis is the singular. Okay, right. Now, it's these metastases that generally kill people with cancer. This is why cancer is so deadly and why benign tumours are less deadly, even though they will go on to become malignant tumours. Okay, generally, it's not the primary tumour that is a problem, it's the secondary tumours that are a problem. 90% of cancer patients die because of secondary tumours. Okay, now why are the secondary tumours so deadly? Well, what happens if this happens in your brain, which is where it generally does happen, that kills you. Okay, if you get these tumours forming in your brain, they're going to destroy neural tissue. Okay, and that's generally what kills you, these secondary metastases within your brain. Okay, 90% of deaths are caused by secondary metastases, and it's mainly secondary metastases going to the brain that is the problem. Okay, right. Now let's talk about metastases to the lymph nodes. Okay, so it's not just cancer cells invading into blood vessels that is the problem. You also get them invading into lymph vessels. Now, lymph vessels are much bigger, much more flimsy vessels. Okay, so we'll have a lymph vessel here. Okay, and what happens then if the tumour invades into this? Well, generally what happens is that the tumour ends up throwing off much bigger clumps of cells. Okay, so remember I told you that when the tumour invades into blood vessels, what happens is that it generally throws off single cells into the blood, whereas when it invades into lymphatic vessels, it generally throws off huge great clumps of cancer cells, okay, into the lymphatics. Now, these potentially can end up going back into the normal circulation, okay, because eventually the lymphatic vessels do drain into blood vessels. However, usually what happens is that they get stuck in lymph nodes, okay. So remember, what is the purpose of the lymphatic system? Okay, the lymphatic system, partially it's to drain fluid from all of your tissues because the circulatory system isn't good enough to do it, but also the lymphatic system is an immunosurveillance system. Okay, basically what is happening is fluid from all of the tissues of your body is being drained in the lymphatic system. Okay, and the fluid, before it goes back into the systemic circulation, has to go through lymph nodes, which are these little offices, if you like, of cells which are looking out for problems, okay, and if they find problems they will launch an immune response to that, okay. So all of the lymphatic fluid is being strained or sieved through these lymph nodes, okay. So if you are bringing back a clump of cells to a lymph node, okay, so if I put a lymph node here, and this is completely out of scale, but if we have our lymph node here, this clump of cells is not going to get through the lymph node, it's going to get stuck in the lymph node. Even a single cell is likely to end up getting stuck in the lymph node. Okay, and then what will happen? Well, this thing of cancer cells, even though it's surrounded by immune cells, is going to make itself at home at in that lymph node, basically, and will start up a secondary tumour. So, you know, this thing will start growing. Okay, you'll start up a secondary tumour in the lymph node. Okay, and this secondary tumour is now known as a lymph node metastasis. Okay, so that's what is meant by lymph node metastases. Okay, right, so I'll just put that here, and then we'll call it there for this video. So this is a lymph node metastasis. In the next video, what we'll do is we'll start off by talking about some terminology. Okay, so we'll talk about what is a carcinoma, what is a sarcoma, what's a leukemia, lymphoma, an adenoma, things like that. Okay, then we'll talk about this pathogenesis in more detail. Okay, so we'll talk about other mutations that are needed. Okay, because we have reduced this down to the most basic scheme that I can imagine. Basically, we've reduced it down to 2 mutations, basically 2 changes. Okay, the initial change causes you to overdivide, then you get the change to malignancy. Okay, and that is good. That's good because it simplifies it down to something very nice and easy to understand, but it's inaccurate. Okay, it's a there are much more steps in between. Basically, it's a process of many mutations building up and up and up, and we're going to go through some of the other ones that are needed, basically.